Okay. A few logistical items. All the resources about this particular webinar, upcoming webinar details, slides, and recordings will be made available at the CGC website. It's the cancergenomicscloud.org website. We will post a link in the chat. These webinars happen every month on the 4th Wednesday at 2 p.m. Eastern. So if you have any questions, please use the chat box for asking questions at the end of the presentation. We will have a question and answer session. And we will also post a link to a feedback survey, which is very helpful for us to collect feedback from all of you to improve the webinars. So please respond to the survey, and you'll also get a follow-up email tomorrow with a link to the survey in case you missed filling it out today. So with that, I'm very delighted to welcome our guests, Dr. Yuriy Gusev and Ms. Krithika Bhuvaneshwar. They're from Georgetown University. They're here to share with us their experiences in using the Cancer Genomics Cloud as a teaching platform for their Georgetown University's Master's in Health Informatics and Data Science program. Dr. Gusev and Ms. Bhuvaneshwar, they are collaborators of our platform, users, longtime users of our platform, and we're very, very happy to have them here. Just a little bit of introduction. Dr. Gusev is an associate professor of bioinformatics and a bioinformatics lead at Georgetown University Innovation Center for Biomedical Informatics, ICBI. He is the director of the graduate program in health informatics and data science, and also co-director of informatics shared resource for the Lombardi Comprehensive Cancer Center at Georgetown. He has several years of experience in academic and industry research in the fields of bioinformatics, systems biology, computational modeling of biological systems, and biomarker discovery. And he has over 20 years of experience in teaching and training in the fields of bioinformatics and computational biology at several academic centers across the country. And he has developed several new curricula for graduate and undergraduate programs at Georgetown. Ms. Bhuvaneshwar is a research instructor faculty and curriculum coordinator for the Master's in Health Informatics and Data Science program. She's also a senior bioinformatician at ICBI Georgetown and has over 12 years of professional work experience and combines her interdisciplinary skills in bioinformatics and biostatistics for numerous projects within ICBI. And she has also helped organize several training workshops at Georgetown. So without much further ado, over to you, Dr. Gusev and Ms. Bhuvaneshwar.
Thank you very much, Sai, for this nice introduction, and also for inviting us and giving us opportunity to share our experiences with using Seven Bridges as a training platform. I'm going to share my screen now. I hope everyone can see it.
Yes.
So today, we're going to talk about how we utilize Seven Bridges platform for training of a new generation of health data scientists. We kind of structured this presentation slightly different than a regular research seminar, because we're not going to talk about our research, but only about the training approaches. So we structured this webinar in two parts. First, I'm going to talk about our background in bioinformatics and data analytics, and our approach and teaching philosophies, if you want, and methodologies for teaching and training a new generation of biomedical data scientists. And in part two, Krithika will present a more detailed information about and examples of how we apply our methodologies in a current graduate level courses, and how we leverage the Cancer Genome Cloud as a teaching platform for our master's degree program at Georgetown University.
I apologize for interrupting. Can you go into presenter mode?
I can try.
Full screen. Yes, perfect. Thank you.
Thank you. So who we are a part of Innovation Center for Biomedical Informatics at Georgetown University. We've been around for more than 12 years now as a team. And we also are part of Biomedical Informatics shared resource for Lombardi Comprehensive Cancer Center. And we've been supporting bioinformatics needs for Lombardi Comprehensive Cancer Center for the last 10 years. And now we also running our health informatics and data science program at Georgetown since 2019. So now we're entering a third academic year of this program. And I believe we accumulate enough positive experience with using Seven Bridges platform so that we can share it with you today. What are our main areas of research? We are not only teaching, we are active practitioners in bioinformatics and data science, genomic data science specifically at ICBI. And ICBI is an academic hub for biomedical data science with a primary mission of conducting investigator-initiated research, as well as education and training and collaborative projects with biomedical investigators, mostly focusing on cancer-related translational research. We also developed and maintained for a long time, over past 12 years, online resource called Georgetown Database of Cancer. It's an open access online platform for translational cancer research. In the past few years, we get involved more and more. We're getting more and more involved in data science and bioinformatics for immuno-oncology applications, and also big genomic data analytics for non-cancer applications, such as neurodegenerative diseases, transplant research, viral informatics, microbiome, and metagenomics. In immuno-oncology, we have a growing number of collaborations in different types of cancer. I put here a list of past and current projects which involve specific types of cancer. And of course, we're mostly dealing with molecular profiling data, multi-omics data types for this type of immuno-oncology research. Over the years, we accumulate a lot of experience doing this type of genomic data analysis. And we notice there is a kind of overarching themes, topics in all of our areas of research. First of all, we are routinely dealing with big biomedical data. And how big is it? This is a question which many students ask us when we start talking about big biomedical data. And actually, it's not easy to explain why we call this type of genomic data, for example, big data. You can wave your hands, or you can try to show it on a computer screen, but it's really not that trivial to understand how big the biomedical data is. And one way to actually make students to understand and learn about big biomedical data is through hands-on experience. And that's basically one of the major approaches we develop over the years to find the way to expose students to actual big biomedical data and learn in their own experience how to handle those data, how to process them, how to do exploratory analysis, etc., etc. But if you think about it, it's not trivial either, because the big data are really big, and they usually start where your laptop stopped working. To really find, it was really challenging to find a right platform where we can implement our idea of providing students with a computational environment where they can actually easily access big biomedical data and work with them. Of course, cloud computing is the solution in this case, and we cannot really avoid it, and it's better to embrace it. And we actually doing it in our research as well as in our training. And the third component, which we always utilize in all the research, is data analytics as applied to data mining for big biomedical data. And this is also a very non-trivial, as you all probably know, even for researchers and professionals, it's non-trivial task to find a way to connect the analytical tools to the big data. The big data usually are located on a cloud, and it's very difficult to download the data, to move them to local servers, where usually you would have your computational tools available. So, of course, the main trend these days is to move tools to the data, not another way around. To have tools, to make tools available on a cloud computing platform, right in the environment where the big biomedical data are located. just briefly touch on big biomedical data origins. Of course, one of the major sources of generating big biomedical data is massive amount of genomic data, specifically sequencing data, genome sequencing data, which are being generated at increasing speed as we speak. We're talking about petabyte, if not exabyte scale on an annual basis right now. Very large projects such as Human Genome Project, TCGA, 100,000 genomes in Great Britain, for example, and 1 million genomes in US is well underway. Some of them already completed, and this actually generate enormous amount of genomic data. There is no doubt that big genomic data really have a transformative effect on biomedical research in clinical practice because they provide a unique molecular level insights in the disease mechanisms and open opportunity to find new biomarkers and new treatment modalities for many human diseases. One of the emerging trends, of course, is utilizing massive genomic data for precision medicine, precision health, to use individual patient molecular profiles to customize their treatments and diagnostic in the most precise way. And to do that, we all need to develop new tools and continue to develop resources which allow to do exactly that, to bridge the gap between the clinical practice and the wealth of molecular data available currently from genomic sequencing efforts. One of the reasons why we see this avalanche of genomic data is, of course, that the technology behind genome sequencing is getting cheaper every year. And right now, I believe we really reach this very desired goal of the cost of a whole genome sequencing per patient is below $1,000 currently. And of course, big data mining is very important. And this field is moving forward really fast. And if you wanted to be up to date with the methods and teach students up-to-date methodologies, we really need to keep up with fast changing landscape of big data mining. Just want to give you one example. This infographics here showing currently listed, currently FDA approved artificial intelligence-based devices in medicine. That means diagnostic and decision-making devices which based on deep learning and machine learning algorithms. There are more than 30 algorithms currently already have FDA approval. And we are in a classroom trying to start to teach students on simple examples how to apply AI in a practice. However, the reality is actually moving forward really, really fast. And it's not easy to catch up with these amazing developments. So facing all of these challenges and having knowledge and practical experience from research, we developed over years our own teaching and training philosophy. We believe that these transformative times in biomedical research and medicine require really adequate approach to workforce training, and most importantly, to teach students how to deal with big biomedical data. There's clearly increasing demand on interdisciplinary professionals who are proficient in both computational methods and biomedical domain knowledge. In our own experience of teaching, we saw two major possible scenarios. We can train biomedical researchers in computational methods, or we can train quantitative science trained professionals to learn about biomedical technologies and data types. It's very interesting that from our experience, both of these approaches actually converge when we're trying to teach specifically topics related to big biomedical data. Biomedical researchers and computational scientists both facing different kind of challenges, but still challenges when they're trying to learn about big data. Because it's really requires this kind of complex multidimensional understanding of the nature of the data, as well as the methods which should applicable to this data. So the ideal, we believe that the ideal new generation of data scientists could be fluent and should be fluent in both domains, computational and biomedical. Just to give you a very brief history of curriculum development for biomedical data scientists at ICBI. We're now entering a second decade of working continuously at Georgetown on developing different kind of training and teaching courses, workshops, and such. We started to develop training workshops for professionals first, utilizing our new online platform, Georgetown Database of Cancer, about 10 years ago. And one of the first experiences we developed a new graduate course in translation bioinformatics, utilizing online platform GDoc to train bioinformatics graduate students. At that point, we already have a platform in hands, which actually allows students to access online open source data collections with molecular profiling data. And we also provided access within the same platform to a relatively simple set of exploratory quantitative analysis tools. By 2014, we also got involved with new curriculum development for undergraduate biology course, system biology, where we utilized a different online knowledge base called Pathway Studio, currently owned by Elsevier. It's one of the premier system biology platform, cloud-based, which allows students to explore a vast knowledge base accumulated over the years of published biomedical literature in a graphical format with network representation of interactions between different types of molecules inside the cells. Again, this kind of, we continue to develop this approach where we were searching and trying to utilize different kind of online platforms where students can log in and collaborate on the same project, do the same kind of task within the same environment, use the same data sets, and apply the same set of tools. And we started to build on this experience, and by 2016, we received the funding from NIH, National Library of Medicine R25 grant to develop a MOOC on EdX platform for kind of develop an online guide and training for biomedical researchers who wants to learn more about big biomedical data. We were combining for this course various online resources, including our own GDoc, as well as TCGA Data Commons, Xena, and other online resources related to CGA. And the idea behind this massive online course was to provide students anywhere in the world with uniform, reproducible, hands-on experience with navigating and exploring big biomedical data. And finally, by 2018, we started to develop new graduate program, which was approved in 2019 and start operating in 2019. And now we're in the third year of operation. I just want to briefly fly over resources I mentioned. This is our Georgetown Database of Cancer. The important feature of this system and platform is that it's study-based. It's based on large sets of molecular and clinical data, and also provide analytical tools accessible on the fly. So we're bringing tools to the data. And it also has this capability of creating user accounts with analysis sessions history, so the students can revisit their previous sessions. And it's also shareable. We allow to have user groups online who can actually share the same project and the same data analysis results within the group. And this is our EdX MOOC available at this link, where we kind of first tried on a large scale develop a set of not only lectures, but also hands-on tools online where we design exercises based on online available data sets and tools, so the students can have the same experience, exactly the same experience from around the world. And finally, Master's in Health Informatics is in-person full-time master program. We currently focusing on accepting candidate with computer science, statistics, and STEM background. It's a one-year accelerated program located at Georgetown. It has really unique program features with a hands-on experience incorporated in every course in a program. We striving to provide at least 50% of each course devoted to hands-on workshops. Our courses taught by well-respected faculty, experts in their field, and it's also a multidisciplinary coursework. This is a curriculum overview, and with the blue arrows, I indicated current 2 courses, which were co-developed by me and Krithika, and both of these courses utilize a 7 Bridges platform. Currently, Precision Health Informatics is a fall semester course. We already did it twice and preparing for the 3rd year in collaboration with team at 7 Bridges. And last June, we use 7 Bridges platform for the first time to teach imaging informatics course. And this is just a brief, short list of main contributors and our advisors from industry government. This is advisory panel for our HITS capstone project. This is a kind of internship project which students must complete before graduating from the program. So with that, I will stop sharing and will allow Krithika to go ahead with the second part of the presentation, and maybe I can entertain a few questions.
Can everybody see my screen?
Yes.
And hear me? Okay.
Yes.
Any questions? If not, I can move on.
We can take questions in the end. People can
Okay.
add their questions to the chat box, so you can go ahead. If you can move to presentation mode, that'll be fine.
Okay, hold on. For a minute, I forgot how to do presentation mode. 1 second.
On the bottom.
Okay, on the bottom. That's why I won't see it. Okay.
Right? Yeah.
No, not. Oh, here.
Yeah.
Yes.
Okay. Okay. Am I good? All right.
Yeah.
So Yuriy talked about our HITS program. So we are a 1-year master's program, and we focus mainly on hands-on workshops and real data examples. And we teach students, help them get comfortable with multiple programming languages, including Python, R, SQL, machine learning, AI, and modern cloud tools. And so why do we use the 7 Bridges platform? The reason why we use this platform, like 7 Bridges for data analysis and also teaching, is because the multiple tools are stitched into one workflow called an app. They're easy to run this app, and you don't need to know about the command line, especially it's great for students. And easy to run batch jobs at multiple samples at the same time. Easy to scale up or scale down based on the number of computational resources needed. You can access the results online through a web browser. It's easy to review and share results, and colleagues can easily reproduce the results. There's no need to babysit the data or the workflow or sit next to a local computer. There have been instances where I have been in the metro, in the train, and I've been able to start batch jobs through my phone. I'll just go home the next day morning. It'll all be done on the 7 Bridges platform. It's very, very convenient that way. And so the CGC pipelines we use in the HITS program include the imaging informatics course, like Yuriy mentioned, where we teach students how to run medical image segmentation pipelines on the CGC cloud platform. We also have another course in fall, Precision Health Informatics, where we teach students how to run NGS pipelines on the CDC cloud. And with this, we are partnering with 7 Bridges, and they're providing free cloud credits for all the students for these 2 courses. So thank you for that. The advantage of using CGC platforms for teaching is mainly because It's a very simple visual representation of all the steps in the pipeline. So it's very easy for students to learn the basics, concepts before we dive into the command line details. And that's great for teaching and training. It's easy to run the pipeline, especially in batch mode. Also easy for instructor to check the classwork and the homework of students, and also easy to teach them about the input files, output files, workflows, tasks, cloud costs. So all components for a data science project is very easy to teach them about all of these different components. So this is how we set up the environment for teaching and training. We have one project for each student, and the instructor is added to each project as admins. We always use public open source data and workflows for teaching, and most of it is on the SB cloud itself. And we teach them how to run the workflows in batch mode, that's parallel processing. And it's also done as an in-class hands-on demo and also as a homework assignment. So here's an example screenshots from the CGC Cloud Platform. We have one project set up for each student. And here's a look inside how the project will look. So this is a student project, and we have the instructors also added as admin. And you can actually easily track and monitor the task that the student is doing and also their homework assignment as well. Students learn the FAIR principles through the 7bridges cloud platform because it's easy to find all the data and the workflows. It's also accessible, it's open and free, it's interoperable and very easy to integrate with other data sources. Also very easy to reuse the data and also reproduce the workflows. I have some example use cases that we use for training. I'd like to share with you. So here's a pipeline that we use for teaching, basically for analysis of medical imaging data. So this is how you probably all know this, but this is how we run a typical workflow on the 7bridges cloud platform. We choose a project first, and then we select the workflow of the app, and then you choose the input files of the data to run. And then you actually run the app itself. So here is an example where we ask a student to type in the imaging word in the text box, and you can bring up two existing workflows, imaging-related workflows in the 7bridges platform. The W indicates it's a workflow. So we ask the student to copy these workflows into their projects, and this is how it would look after they copy the projects, the workflows into their app. And then we also teach them how to query public data sets through the query browser in the 7bridges platform. Here is an example where in this example, we looked for medical imaging, that is MRI files for one patient from brain cancer from the TCIA data collection. So through this experience, the students will also learn about big public data collections like the TCGA or TCIA collections. And they also learn how to query and look for what kind of data they're looking for based on a specific query. And here is an example where we copied, based on the query, we copied all the input files into the files folder inside the 7bridges platform. We can also easily see the metadata here. So it's very convenient and easy to understand that way. And here is an example where we run the actual workflow itself. Here we turn the batch mode on, so it's very easy to run the parallel processing. And then here is a simple workflow which will uncompress and view the MRI images. And you can just click on the run button and it will run the workflow. And again, it's easy to, these are all different child tasks which will be initiated for every batch job that's run. For example, if I run a batch job on 8 samples, there'll be one child task for each sample, which can be easily seen here. You can see which are running, which are completed. And here, once the workflow is completed, you can actually click on a task that's completed, and you can see this is the input file, this is the output file. Then click on the output files in these hyperlinks to view the output files and how they look. And you can see the cost was about 0. It was free. It was so cheap. It was free, and the runtime was less than 1 minute. So here, this is the example end-to-end workflow where we chose the project and you choose the workflow and you select the input files and you run the app. So at the end of this class, the students are able to learn what workflows are used for medical imaging for this specific use case, and what kinds of open source workflows are available, what kind of public data sets can be used for this kind of workflow, how to run them in a batch mode, what the expected output is, and the cloud computing costs and the runtime. A second use case is how to run a Python notebook on the 7bridges CGC Cloud Platform. We work with David Robertson. A shout out to David if you're here. And he demoed a real life example of COVID-19 lung segmentation using deep learning. So how this environment was set up was David shared a Jupyter Lab notebook with us, with all the students. And the environment was a machine learning environment which had TensorFlow installed in it. And we used GPUs to execute the code itself. And you can see it's not that expensive to run on the cloud, especially in the 7bridges platform. You can see it's all self-contained. So all the files relevant for a student project was all inside this one project, inside this project. You can see the data, the Python notebooks here, the models that were used, and also the slides are also here. So it's self-contained. And this is the notebook itself. At the end of this class, the student got an experience as to how to run using command line specific code on the cloud to run a specific, for example, in this use case was lung segmentation using deep learning models. And they also got experience with GPUs and TensorFlows in the CGC cloud. And they were also able to understand what the output was, what the cloud computing costs were, about the data and the algorithms. So basically all aspects of a data science project is very easy to not only understand, but also teach these concepts to students in a master's program. An example use case 3 here is the Genomics Pipeline. With Genomics, at least in our experience, because it has multiple steps in the pipeline itself, is a little bit intimidating, maybe perhaps for students to learn for the first time. So by using the CGC platform, all the steps are all very visual and also in different modules. So it's very easy for them to understand and understand the concepts of the NGS pipelines itself. So here is an example of a project where we use RSEM workflow for RNA-seq alignment and quantification. Here is the workflow itself. As I mentioned, you can see it involves multiple steps and is having a visual representation on the CGC platform is very easy for them to understand and also not only understand, but also for us to teach as well. So the input files for this workflow, we input FASTQ files and the genome reference files and the information about the gene and the coding regions. And the output file, the tool itself will run alignment and quantification. And it's also visible here. This pipeline, we modified and customized this workflow with the help of the 7bridges team for teaching purposes. So this is how we run the workflow. The workflow is run on the batch mode. So once we identified what input files we want to run, the batching is turned on so we can run it in parallel. All the samples are run in parallel in batch mode. So this is the GTF file, which has information about the gene and the coding regions. And then the input files are here. This is, you choose the reference file and then you run the workflow. Again, here, this is a full excellent snapshot of what the workflow looks like. So this is the input file. It shows you what were the input files, what were the settings of your tool, and what the output files were. Here, in this example, you can see output files from alignment and also quantification. And it's also an excellent summary of the cost, which is very cheap, and the duration, which is about 30 minutes because we use spot instances. The runtime will depend on the size of the VM. So by using these this sort of built-in workflows, it's much easier to teach students that by changing the size of the VM, the cost would change, the duration would change. So various aspects of a data science project in real life, it's easy for students to get first-hand experience about these things. Here's an example of the DNA-seq pipeline. Again, here, this is a little more complex than the RNA-seq because it has not only quality checks, we have sorting, we have alignment, and then we also have the gene annotation. So multiple steps are there as part of this pipeline, and having a visual representation it makes it much easier not only to teach but also to learn. Here's an example of our file, how the file folder is set up. And the concept at the end of this class, students will learn basic concepts about NGS data analysis, how to run RNA-seq whole genome and whole exome pipeline on the CGC cloud platform. We'll also understand what the input files are, what the output files are, the cost for running these kinds of pipelines, and also file sizes for genomics data. As you know, with the file sizes for a whole exome is much smaller compared to RNA-seq, which is more medium-sized, and whole genome sequencing is very big, based whether it's clinical depth or not. So all of these concepts is very easy to teach and also learn with the help of the CGC platform. Yeah, overall, we've had very positive feedback from the students because they were able to focus on the concepts instead of worrying about setting up the environment or versions, or if you have a different operating system, that you can have challenges with respect to installation of those tools, setting up your shell scripts perhaps, and also cloud costs, or even if you're doing it on your own laptop, which is not really feasible for whole genome sequencing data. It's very time consuming if you want to use your own personal laptop, but it makes it much easier and much easier to focus on concepts if you have a platform like the CGC platform. So in summary, in our experience, we found 7Bridges platform a great tool for teaching, especially the concepts of cloud computing, because the data and the tools are already there. And all we have to do is teach the students what the pipeline is about and what the pipeline does, what the input files are, how the output files should look like, and all about scalability and cloud computing costs. And it's also very easy as the instructors, it's easy to manage and monitor what the student is doing, not only in class, but also as a homework assignments. And students are able to reproduce the in-class work at home. At the end of the course, at the end of the semester, we ask students to keep copies of their work so they can continue working on their projects after the course is done. Any questions here? I'm going to stop sharing. Yuriy, do you want to add anything?
Maybe just on student feedback, I just want to mention, we forgot to put it on slides, that some of our students continue to use 7Bridges platform beyond just training environment, and at least 4 students were using it for our summer internship project with industry partners, successfully utilizing the skills they learn in a classroom for real life projects. And one of the students also successfully utilized his experience with 7Bridges to land a job, where he actually it's a really interesting success story where the student was able to incorporate in his online interview a demo which he provided on the fly, a real time demo of how he managed genomic data analysis on a 7Bridges platform. And this probably was one of the key components of his successful recruitment to one of the major medical research organization in the country. Thank you for your attention, and we are ready for your questions.
Great. Thank you so much, Dr. Gusev and Ms. Bhuvaneshwar. This was very enlightening. And what makes me very happy is, NIH makes so much investment on basic and translational research, and there's a lot of data that's available. And you rightly pointed out that all of this corpus of rich data can be used for teaching purposes and training the next generation of data scientists. So that's incredibly powerful on its own. And I'm so glad you're utilizing the power of the cloud and the platform for this kind of research. And so with that, I would like to thank our speakers who introduced this concept of how the cloud-based platform can be effectively used as a training and teaching platform. So we are open to taking questions. So please feel free to post in the chat box.
And once again, I want to thank organizers for this opportunity. We love what we're doing, so we love to share our experiences. And I believe there is a lot of room to use 7Bridges for more training and more teaching. It's really almost an ideal platform.
That's great to hear. And we would like to incorporate all the feedback and make it better and more user-friendly for the training programs. While we're waiting for others to post their questions, so I was just wondering, can you share with us what are the next steps? Like, are you going to be using the platform for other kinds of teaching courses? Can you share with us your future plans for teaching?
That's a very good question. Krithika, do you mind if I'll start?
Go ahead.
Basically, you know, we're constantly thinking about expanding workshops with hands-on experience, and of course 7Bridges provide a great opportunity to do exactly that. We start to think that for these two courses where we already utilizing 7Bridges, we might find a way to expand examples and use cases for students, for example, with single cell sequencing. This would be one natural extension of what we're already doing. And the tools and data, my understanding, are available at 7Bridges. And right now, we only provide them as a topic, as advanced topic with lecture only component, but we would love to add a workshop component where they can actually try, I don't know, maybe a Serat package on 7Bridges and see and experience firsthand what does it mean to analyze single cell sequencing data set, which is quite different from other sequencing data types. For imaging, the same. I think as long as the 7Bridges team will add more material and more pipelines to imaging part, we would be happy to expand it. And another thought which I had, that we have this spring semester course on AI applications for health data science and for machine learning and deep learning applications. And I'm very curious to explore how we can find a way to use deep learning tools available through 7Bridges platform as an illustration to this AI course. That's kind of immediate thoughts about it. Krithika, do you want to add something?
Yeah, we also have another advanced module in our HIDS 503 Precision Health Informatics class, where we teach students about the basics of metagenomics and also viral informatics. So we already have those pipelines already inside the 7Bridges platform. So also another natural extension would be to incorporate those pipelines into our course. Also, recently, I know 7Bridges has integrated Python and R Studio modules inside the CGC Cloud platform itself, and we're already using the Python modules. But especially for RNA-seq pipelines, to teach students how to run differential expression analysis, we could easily use the R Studio version directly to take the gene counts files from there, and then all we have to do is import the data with our scripts into the R Studio Cloud platform and easily run all of our differential expression and analysis on the CGC Cloud platform itself.
Yeah, that's a great idea.
Great. Yeah, I think these are pipelines that are already available, and I like the ideas you shared, and we'll be more than happy to help structure the course, but also add new tools or workflows as needed for future classes or symposium and all that. And I'm going to be sharing about a symposium that's coming up in August, which covers a couple of topics that you already mentioned. We have a topic on single cell analysis and also on machine learning and epigenomics as well. So there's one more question I wanted to ask before I move on to the chat. So I know you were talking about how effective the platform is. I just wanted to get a sense of what do you think are the barriers for existing teaching or training programs to transition to the cloud for teaching purposes? I know the way you described, you were familiar with the cloud platform and it seemed almost easy, but I know there are probably barriers. And did you have any challenges, or are there any challenges you think are out there that people are not able to immediately transition to using the cloud?
I think there are some barriers. First of all, there are many systems, commercial systems available, and we actually had challenges to find the right one. Most of the systems face this problem with scalability. And for cloud computing platform for genomic data specifically is a key component. If the pipelines or platform is not scalable to large enough data sets, it's not usable, not for teaching, not for research. So that was one of the challenges to find the right platform. There's another, of course, obvious barrier for academic teaching is costs of commercial platforms. And I must say that 7Bridges is really stand out as a platform which basically sponsoring our academic efforts in teaching. And I think this is a way to go, because by training our young generation of data scientists on 7Bridges platform, we not only provide them with up-to-date experience, but also those are potential users in the future of the platforms. So I really would encourage your team to consider continue to sponsor academic efforts in teaching specifically. Maybe think about the student version, student access, special type of student access to the 7Bridges with some limitations perhaps, but sponsored by the company, because we need to continue this training, and I think 7Bridges could be a very effective way to train new generation of data scientists. But to do that, we need to remove the financial barriers.
So outside of the 7Bridges platform, if a researcher or a trainer were not using the 7Bridges platform, if you want to teach or even do research, you have to set up the cloud environment on your own. So basically like Amazon or Google Cloud or whatever cloud platform that you use, the researcher has to set up the environment on their own, install all the tools, make sure the tools are compatible with the operating system. So those are all challenges that the instructor or the researcher would face before they even start doing the work itself. So installation, all of that takes time, and it takes several days to just get all of the tools installed, get the environment set up, especially for training purposes. We know of other teachers who teach other aspects of genomics, but they have like a VM that they have to replicate for each student. It makes it more complex for teaching purposes itself. But that's why we use the 7Bridges platform. That way it's much easier, and all of those technical components are removed, and we can solely focus on the teaching of the content itself.
Got it. Yeah, and thanks for that feedback, Yuriy. So we are also closely working with our funding agencies to support training on the cloud platform, and we are working on some ways to support the training by covering the cloud costs. So if there are others in the group in the webinar today who are interested, feel free to reach out to us. So we have a couple more questions in the chat. So the first question is about, I'm just starting using 7Bridges, what best place to learn by example? So I just saw Manisha has posted the link to the 7Bridges Cancer Genomics Cloud documentation. And there are lots of tutorials available and also guides that are available. Another question from the same attendee is, are there pipelines using single cell data, RNA-seq, proteomics, epigenomics, mutation annotation, and clinical data? So there are several pipelines. I can just quickly answer that. There are several pipelines. You can look at the documentation. There are several existing apps and workflows. So you will find them in the docs pages. And if you still have more questions, feel free to reach out to us. We have another question. Great presentations and examples for teaching primary analysis of the genomics data set. Did the platform also provide students the opportunity to implement and optimize tools in the cloud environment instead of relying on vendors? So I think instead of using them that are already available. So I think the question is mostly, did the students implement their own tools and optimize them?
Right. If the question is about the 7Bridges platform, then yes, we for our research we usually start with available pipelines, but we modify and optimize them to our needs. And of course, this could also be used for training. We have multiple use cases based on our research experience and published results where we can implemented for teaching. In terms of our program approach, yes, what we described today is just a usage of 7Bridges. Other courses rely solely on direct usage of cloud, for example, for artificial intelligence applications, Google Cloud mostly. So we are teaching students how to go beyond prepared pipelines on a cloud, on a Google Cloud environment in our case, because Georgetown primarily focusing on Google Cloud. So there's two parts to this answer.
Great. Thank you. We have one more question. Can students perform cohort mining such as TCGA, Target, etc., and correlate genome omics data with clinical pathological information like age, sex, survival, etc., for biomarker discovery for early diagnosis and prognosis markers for specific disease and race types? Yuriy, do you want to take it? Then I can add on to that later.
I can mention. Technically speaking, of course, this is possible. And in terms of the students' involvement, Krithika mentioned one of our planning development is to utilize most of what was described in this question usually done using different kind of analytical approaches. For example, in R environment, that's what we do. The correlation analysis we do is done in R. And because R Studio is available through 7Bridges platform, we are thinking and planning to implement the same scripts which we use in the classroom on the local machines. We would prefer actually to have developed these modules in RStudio environment because the data are there. And the question, I like this question because yes, it's correct. The TCGA and target data, especially process data, are available without restrictions. So it would be very convenient to do this kind of correlative analysis on the fly on 7Bridges platform. And I believe many people actually do that for research, but for teaching, that's something which we are very interested to develop.
Yeah, I want to add that it can be done. It can be possible in the 7Bridges cloud. You can use the data query tool to query the TCGA or the target cohorts to identify which patients that you want to use for a case and control groups, and then you can use your RStudio scripts to run the analysis inside the SB platform itself.
Great, thanks, Yuriy and Krithika. And just want to add on to that is, if you're a student or a postdoc and you are interested in doing these kinds of interesting research, then we also have collaborative awards. So you can reach out to our team. There's this email, you can reach out to us, and we'll send a form. It's a cloud credit that you get for performing analysis on the platform. You get $10,000 worth of award for these kinds of cool projects. So feel free to reach out to us if you have such specific research problems. So we have Dave and Manisha responding to the questions in the chat. I think this was really great. I just wanted to share a couple of help information. So there's the Knowledge Center, which has documentation. And if you want to reach us, you can reach our support team. And we also have regular weekly office hours that happens on Tuesdays and Thursdays. So all you have to do is go to the website, and then in the help section, you'll have office hours. You can find the links to join. And I have a couple of announcements. We have two major events coming up. One is a summer symposium, which is going to happen on the 18th of August, which is a Wednesday. And it's a 2.5 hours. So it's supposed to be till 2:30 p.m. actually. So it starts at 12 p.m. Eastern and ends at 2:30. And we have 3 sessions. One is on image processing using machine learning. The other one is single cell analysis. And we also have epigenomics. So very exciting topics. So our team of 7Bridges experts, you can see them all here in this image. They will be presenting on these topics, and also a live demo of how these tools and workflows can be used on the 7Bridges platform. So that's a symposium that's coming up. So you can register in the 7Bridges CGC website. It's in the events page. And we will also post the link for this in the email that you'll receive tomorrow. And we have the August webinar that's going to be presented by Margaret Lignan. So she's a bioinformatics consultant and also a collaborator of Seven Bridges CGC for a long time. So she's going to be talking about how she has been using the CGC for exploring pan-cancer dysregulated pathways and identifying epitranscriptomic patterns in the TCGA data. So that's going to be an exciting topic. So if you're interested in this topic, please feel free to register as well. With that, I think we don't have any more questions, but we do have some chat going on in the chat box in response to the user questions. I would like to thank Dr. Yuriy Gusev and Ms. Krithika Bhuvaneshwar again for joining us today and giving this wonderful presentation on how CGC has been used for their training programs.
It's been our pleasure. Thank you for inviting us.
Thank you.
Thank you. Bye.
Bye.